today's talk, I'm going to first just give an overview of what kind of research questions we're interested in, and what is our current existing analysis pipelines that we're using, and lastly, our current development of machine learning tools in that to help us reconstruct the neural circuits, and that is in collaboration with DR Vision here. So I also prepared 2 demos, and that I will be interactively in and out from my presentation to the demo. So here, can everyone see my slides? Okay.
We can just see your title slide.
Okay, so it's moving now, right?
Okay. I'm seeing a blank screen, Wan-Qing.
Yeah,
it's just black.
Yes, so it's fine.
Okay.
And the brain is constituted of millions of neurons, and they form subtypes that bear unique shapes or connectivity and functions. So the synaptic connections between the neuron subtypes are very specific and often needs to be refined during development. For example, here this is gray neurons at maturity maybe connects preferentially to the magenta neurons in a stereotypic ratio. However, in the development, they need to also connecting to other cell types. Our lab is interested in understanding how such precision in synaptic wirings established during development. And we are also interested in how circuits disassembled in the disease conditions. So when some cells die, how do the remaining neurons respond to the loss of the synaptic partners? And furthermore, and if we want to repair the neural circuits, we would also need to know how well the regenerated new cells or transplanted new cells integrate into the remaining circuits. We are addressing all these questions using the vertebrate retina as our model system. And these beautiful images of the mouse retina was taken by a former postdoc in our lab, and it illustrated a complex but well-organized layers of cells and their connections. So the light evoked signals is conveyed from the rods and cone cells to the retinal ganglion cells, through which is the output of the retinal output cells of the retina, and it's through bipolar cells, and also the signal transfer was modulated by the horizontal cells and the Wan-Qing cells. So the laminar arrangement together with its diverse cell types make the retina an excellent model for investigating the assembly, disassembly, and reassembly of the neural circuits. In order to study the neural circuits, we actually need to reconstruct the cells and their connections in 3-dimensional. And we also need to be able to visualize individual synapses, which are less than a micrometer. The electron microscopy is still necessary to review these synaptic connections at ultrastructural resolution. So several semi to fully automatic methods to acquire EM volumes are available. And the ones we are mainly using are actually called serial blood phase scanning electron microscopy, which was developed by Winfield Deng and Hans Holzmann in 2004. Actually, you already seen this video clip earlier and by Carlos. This video clip is actually from Zeiss microscopy, demonstrating how the serial blood phase EM works. The microsome is mounted inside a microscoped vacuum chamber, and the sample is positioned on the specimen holder. A diamond light removes the thin layer of the block face, and after each section, the exposed block face is imaged. The sample is then moved up a small step, usually for us it's 50 nanometers, and then the sectioning and imaging is repeated. And in this way, hundreds or thousands of images can be obtained and subsequently reconstructed to create a 3D volume. Currently at UW, we actually have already 3 SEM systems and to Zeiss reviews and April from Thermo Fisher Scientific. And typically in our lab, we actually acquire images at 5 nanometer in XY position and cut at 15 nanometer thickness. We acquire not only one single tile, we actually acquire multiple tiles usually, and to cover the region of interest. Each tile is usually 8,000 by 8,000 pixels, which is about 40 micron by 40 micron. And we typically cut hundreds to 1 to 2,000 sections, which can take several weeks or even months. So which depends on the size of our eye. And the whole data set is usually ranging from like hundreds of gigabytes to a few terabytes. First, I just briefly show our current existing pipelines, and which is very labor intensive. And the first step of the image analysis is to do pre-processing using ImageJ, which includes the converting the file format and bit depth and also adjust the contrast. And we then will create a 3D volume from individual 2D images. We also using TrackEM2 plugins in ImageJ to stitch and align all the image tiles. And this is just showing an example of the single section of stitched images from 3x3 image tiles. Usually using the rigid transformation, the TrackEM2 can perform fairly well. And however, occasionally there is a misalignment at the overlap regions. And you can actually improve the registration or stitching by fine-tuning the parameters provided in TrackEM. But it is actually, you probably need to do iterations of stitching, and which will take usually like weeks, depending on the data set you are having. Once the images are aligned, we reconstruct circuitry and manually at present. And this is just includes 3D neuron tracing and subcellular structure detection and also boundary segmentation. Here is just a video clip to show how we are manually reconstruct. And we first just create a skeleton of the neuron of interest. We need to make sure that we are following all the branches of the neuron, because that's not just a single straightforward tree structure. And then we need to manually segment the cell membrane. And this is actually very time consuming. And basically we are painting the neuron section by section. And also depending on the size of the neuron, and tracing a neuron can take like a few hours to a few days. If you need to fully segment or paint a neuron, it takes a few more days. And if you also need to annotate all of the synapses of the cells, that will add up additional day or two, and even a few more days if the cells are really big. We then export out all of our 3D reconstruction and import to Amira to prepare images or the videos for publication or presentation. So there are a few challenges during this image analysis. And first of all, and that's very labor intensive, and we are actually having a lot help from the undergraduates and annotators to help us paint. And second, we also need to navigate through multiple tools and the software environment. And third, and currently, because of those time-consuming process, we can only analyze a limited size of the neural network. Also, we cannot really easy to register the light microscopy and the EM images together. I'm going to dive into our current development. And I want also to mention that there are definitely there are other tools that's available first besides the TrackEM for annotating the neural circuits. And here, this is a really good chart comparing all of the tools that's available and in Daniel Berg's paper. Daniel Berg is actually a developer of VAST, which is listed here. So like what Sam mentioned before, and some of these tools can just provide solutions to address one or two challenges that we are facing. And also most of the tools listed here do not have the capacities to automatically segment the whole neural data set. There are also great efforts in the community towards analyzing large-scale connectomic. For example, there are the Fly-EM project in Genea Farm that's also collaborating with the Google AI, and also the Microns program, which focuses on the mouse brain visual cortex, which is the collaboration work among Princeton University Allen Institute for Brain Science and the Baylor College of Medicine. They are also publishing a lot of the open source tools that can be used for stitching and segmenting, and also then automatic detection for the neural circuits and also synapses. But those tools, first of all, they're like very highly specialized, and which requires we have a very good like software engineer to help us to really connect all the tools together. And also the deep learning work, either machine learning tools or the deep learning tools they are using, they are already trained for their specific data sets. And if we want to use it, we still need to retrain the model and for our specific data set, which will acquire like high performance hardwares to train those networks. In the small labs, and we don't really have the capacity access to those really high-end hardwares. And so with all those limitations, we are actually starting to collaborate with DR Vision to develop intelligent connectomic analysis tools. We are trying to develop a tool that's providing a uniform platform to really analyzing the to use the machine learning to speed up reconstruction, and also to support the whole image analysis pipeline. And also we wish that these tools can be available and easy accessible for a lot of labs. So here are some of the image analysis components I already mentioned that we are going to include in this tool. And besides the component I already mentioned, that we are also developing tools to generating the neural circuits, and also to register the LM and EM images together. So today, I think I'm going to focus on the boundary segmentation, 3D neural tracing, and the LM-EM registration model. I want to say first that we started this collaboration with DR Vision last year. And so this is just a 1-year effort, and we are just prototyping different tools at the beginning at this moment. And so all the results are preliminary, but we have already exciting progress to share with you guys. So the first thing is that we are actually using deep learning model to automatically detect the cellular membranes. And this UNet, specifically 3D UNets, that's the ones we are using. And that's been already brought up by many of the presentations beforehand. And so with this deep learning convolutional network, we're just creating the ground truth for some our subvolume of the EM images in our lab, and then we're providing this ground truth to DR Vision, the development team, and they are training the 3D UNets and to generate the confidence map, and then after post thresholding and post processing, and we're generating the segmentation results. So the deep learning training was performed and using the TensorFlow Keras on a computing platform with 2 NVIDIA GeForce GPUs. And a typical deep learning training run took about 10 hours at the moment. And here I'm just showing results, just using a subset of data annotated, we can achieve a very satisfactory boundary segmentation. So far, we are also testing the trained models on the subsets to scale up to the larger volume of the data sets. And also, we are trying to apply transfer learning to use the trained deep learning model to segment other data sets than what we are requiring to train the model. The next component of where we can do the semi-automatic segmentation, but of course we would also like to have a fully automatic tools to help reconstruct neurons in 3D. And we are developing a model based on fully segmented EM images. And the first step of the model connects the small objects by combining the small masks from a Z slice to its neighboring Z slices. And the second step is connects large objects by combining the large masks from a Z slice to its neighboring Z slices. The last step conditionally merges small objects with large objects and then perform the 3D connected component operation to generate the final 3D objects. The mask association rules are also based on the Jacob index and an overlap ratio of the mask neighboring Z slices. And we are still improving the algorithm to achieve the better performance and scaling it up to apply to on a large EM volume. Here I'm just showing an overview. This is the one of the reconstruction tracing results from a testing dataset, and this is an individual profile. The left side is ground truth that we painted in the lab, and the right side is traced by the algorithm. And you can see this is very good tracing. And sometimes we also can see that the traced cells missing the portion, small portion, and usually those are like thin processes from the neuron. This is basically also due to the segmentation results. The segmentation results sometimes will miss segmenting the small, very small and thin processes. So we are actually going back and forth, improving both the segmentation algorithm and also the 3D neural tracing algorithm to somewhat improve the tracing result at the same time. So finally, we are also combining the SEM with the light microscopy, largely because using the LM, we can actually identify and target cells of the known structure and function for reconstruction. The one reason to do so is to determine whether two neuronal cell types receive similar or distinct connections from one type of the presynaptic cell. So in a project that we collaborated with Greg Schwartz's lab at Northwestern University, the two adjacent retinal ganglion cells with overlapping dendritic arbors was intracellularly diffused with Alexa 480A, which is green here, and in a transgenic mouse retina, in which one of the presynaptic bipolar cells were expressed red fluorescent protein, T tomato. So the light responses of these two cells were also recorded. And after that, they will fix the tissue and for EM. And also, in order to relocate diffused cells in the EM volume, fiducial marks were burned into the tissue using near-infrared branding technique, essentially by line scanning at high power using a 2-photon laser. So the retina was then processed for serial block phase SEM. And we sectioned and imaged the tissue volume of about 150 by 160 by 70 micrometer. The reconstructed skeleton for both retinal ganglion cells confirmed that we successfully identified labeled cells in the LM. And we also could confirm a synapse in the EM reconstruction, which is indicating by an arrow here. And between the ganglion cell and the specific bipolar cells labeled in the TD tomato expression, and then also the LXI488 in the EM images. But this registration is mainly manually, and I have to just comparing the EM images and the LM images side by side. So it would be great if we could register the EM volume to the LM volume or vice versa to speed up the claim analysis. So as such, we are also developing a semi-automatic image registration tool for claim analysis. So briefly, the corresponding landmarks were manually identified at the dendritic branching point of the label cells M and the EM stacks. And the landmarks pairs were used for rigid registration process to derive the transformation matrix. The LM stack was then transformed and scaled so that it can be superimposed with the EM volume at high resolution. And with registered claim images, and we could quickly identify a synaptic site of interest. The white box shows here a potential synaptic site between the fluorescently labeled bipolar cell and the dye-filled ganglion cell, which was localized upon examining the Z stacks of the LM images. After we are switching to the registered EM images, we could easily confirm that whether this is indeed a synaptic site by looking at the ultrastructure. I also prepared a live demo for this section, but I'll go back to that at last. So just a summary that we are using the serial block phase SEM generate 3D EM volumes at a nanometer resolution to reconstruct the neural circuits in the mouse retina. However, currently the circuit reconstruction is very labor intensive and very limited. And we're currently developing an ICA platform to speed up the reconstructions, and also developing tools for claim registration analysis. Although we are currently just using a smaller neural circuits, analyzing small neural circuits, but we are also aiming to scale up in the near future. And with that, I'd like to thank my PI, Rachel, and all of the lab members in the lab, especially our EM specialist, Sean, and all of our collaborators at DR Vision. And also thank you for your attention. And I will restart Avia, and hopefully I can make take a successful demo. But while we're waiting, and I can take any questions if you have.
Hi, Wan-Qing. I'll read you the question since you're busy setting this up.
Yes.
So one question from Shasha here. She's asking, how many samples did you use to train the pixel classifier for the EM image? And were you able to use different type of models for the training?
We are actually using right now we are actually using just one data set that we acquired for the project and to train the UNet deep learning model. We're definitely trying to, as I mentioned, we are actually trying to use this trained model to apply to other data sets we already have. Like the other data sets, not only for the mouse retina, we're also going to try in the zebrafish retina, and also the next year, we are going to expand to apply the model to other areas, other central brain areas in different animal model. So let me try this demo again. Hope it worked. Okay. I'll find another cell. Enable painting. So here on the first section, I just draw the outline of this cell. And actually, this is already jumping to the next section and already paint on the whole, fill the whole cell for me. I'm coming back, so it will automatically fill the cell after I draw the contour, and then jumping to the next section to predict what I'm drawing from the trained EM model. And then if I'm just stepping to the next session, it will start to draw automatically for me. And you can see that this segmentation looks pretty good. And even at this location, there's like already blurring area. It is still pretty okay. And this process is definitely disappearing or moving towards. Yes, and it handles beautifully here. So whenever, I just want to say, whenever we need to do some corrections, it's available and just can clean the segmentation results very efficiently, and you can just step through. At the end, usually at the end, it's like you will getting kind of less confidence. While it's disappearing, it will start to picking up other things. And after you are done with this one and the cells, you can actually complete the mesh. The cool things is that you can immediately see the 3D structure in Avia. That is very convenient. And I have other things painted on this section. So hopefully this is a good demo to show. The next one I want to show, if I have time, I will show the EM.
Go ahead, Wan-Qing, you have a few minutes.
Okay, great. This is a bigger data set, so it will take some time to load it out. So here we actually have the registered EM and LM images, and the green channel is the ones that we are dye fill the ganglion cells process, the dendritic arbors, and then the red ones is the fluorescent label, the presynaptic bipolar cell type. This is still not like the whole volume that we are taking, it's just subset of the volume. So you can see with the sparsely labeled LM images, I can quickly go to identify, find some of the potential synaptic site by trying to locate a site that you can see the green fluorescent and the red fluorescent like overlap. So this looks like a good potential location. And then I will turn on EM images. This will load a little bit. Okay, so at this side, the green is actually you can already see through the LM channels. The green and the green profile, the EM is showing a big process, and that is actually a ganglion cell process that I can see. The red at this section, it is not completely showing a registered button, but let's move up a little bit. Yeah, while you are moving the Z section, you can see the green fluorescent going that way. It's actually aligned pretty well. The red one, buttons becoming bigger now, and also registered to a button profile, which is a presynaptic bipolar cell. So I'm going to turn off the LM images in order to show the details of the EM here. Okay, this is the part it will use a lot of my power. Okay, so this is the profile of a bipolar cell that I know of. And then this part is the ganglion cell process. So this is the green one, this is the red one. And then as I'm moving a little bit, so did you see this like bar shape, a dark gray bar shape? This is called ribbon. It's actually an indication of there's a synaptic connection. This is a synapse. So there's actually a beautiful synapse between the ganglion cell and the bipolar cell. So in this way, I don't need to go through the whole stack to trace all of the synaptic arbors, and then to trying to identify whether this button is labeled in red in fluorescent images. And I just need to seek from the LM to look for potential site and then go into that site to confirm whether that's a synapse. I hope you can appreciate how easy that would help me. And with that, I think I'm going to end my presentation.